I'd like now to get us going on the next session, which is modeling capabilities with Serosurveillance data. I think I was already introduced earlier today, so I won't introduce myself again. And we have three excellent panelists for our last session of the day. First of all, I'll introduce the three panelists, then we'll start off with our first speaker. So our first panelist, who was actually given a little bit of a shout out in the last session, is Dr. Sang-Woo Park, who is a fifth-year PhD student in the Department of Ecology and Evolutionary Biology at Princeton. And he's led the COVID-19 response modeling on campus at Princeton, providing model-based validations of testing and isolation guidelines. And his research now focuses on developing new mathematical and computational approaches for using serological data as a surveillance tool. Our second panelist, who you've already met, if you were here yesterday, is Dr. Carolyn Wagner. Yes, please come join, come up to the front, except for, of course, Jane, who's online, but we can see you online, Jane. So Dr. Carolyn Wagner is an assistant professor in the Department of Bioengineering at McGill University, and she completed her PhD at Massachusetts Institute of Technology in Mechanical Engineering, specializing in the characterization of complex biological fluids, and did her postdoctoral training at Princeton University in the area of mathematical disease modeling. She's a HEMANET member and part of the network's cost effectiveness working group. So welcome, Carolyn. And then our third panelist is Dr. Jane Heffernan, who is a research chair and professor in modeling infection and immunity in the Department of Math and Statistics at York University in Toronto. She is co-director of the Center for Disease Modeling, co-lead of the Canadian Immunization Research Network Modeling Group, scientific advisor to CITF, and is current president of the Society for Mathematical Biology. So a lot of hats to wear. Dr. Heffernan's research responds to the pressing need for new mathematical and computational methods of mapping, understanding, and controlling infectious disease in individuals and in populations, so within host as well as population modeling. And her research aims to influence the development of new medicines and vaccines as well as evidence-based public health policies. So we have each panelist will give a presentation, and then we should have at least 15 minutes or so for questions at the end. So I'd like to ask Dr. Park to begin. And then we'll go from there. Dr. Park, the floor is yours.
How do I...
This little clicker should advance for you. I think the arrow moves it ahead.
Hello. Is this mic working? Okay. So today I'm going to talk about how we can use serosurveillance data to clarify epidemic trajectories. And I'm going to use Enterovite C68 as a case study to show how serosurveillance data can help us better model. Background, Enterovite C68 is just one of many enterovirus serotypes which is responsible for many childhood infections. Particular Enterovite C68 was first discovered in 1962 and has been mostly associated with mild respiratory infections. So since its discovery was largely unnoticed and very rarely reported until 2014, when we had our first large outbreak in the US and many other countries. And since then, outbreaks were reported every 2 years in the US. One of the major questions that we had was whether we're going to see another outbreak in 2020. And besides the respiratory symptoms, another main reason that people were interested in studying the dynamics of Enterovite C68 is its causal implication for acute flaccid myelitis, which is a rare neurological disease which also emerged in 2014 with polio-like symptoms. And there's been lots of laboratory, clinical, and some modeling results which indicate that Enterovite C68 is likely to be a major cause of oxidative agent of AFM. And so the big question being whether can we predict Enterovite C68 outbreak in 2020, and so whether we can predict an AFM outbreak in 2020. So to model this kind of dynamic, the simplest way we can go about it is use what's called the SIR model. So the model assumes that this is the simplest mathematical model we can use to model population level dynamics of infectious diseases. And the main assumption is that everyone's born susceptible, and that once you come in contact with infected individual, you become infected, and once you recover from infection, that you're fully immune for the rest of your life. And one of the key parameters of this model is called R0, or the basic reproduction number, which is defined as the average number of secondary infections caused by each infected individual. So let's say I come in with Enterovite C68 here, and none of you have seen Enterovite C68 before. Then am I going to infect 5 people or 20 people? And that tells us about how transmissible the virus is, and that's going to tell us how much, if we were to develop a vaccine for this particular pathogen, what proportion of population that we're going to have to vaccinate in order to prevent the disease from future outbreaks. And that's also going to be related to what seroprevalence curve we see, because we're going to see later on. And it's we find that SIR model can capture the dynamics of enterovirus C68 pretty well. So this is data coming up from a company called BioFire, which sells multiplex PCR kits to different hospitals, and the results gets uploaded to cloud. And we were able to get the data with collaboration from them and New York, Ohio. We had a pretty good match, and Missouri and Utah, Colorado. We can also see the green, no, not that's orange, yeah. Orange plot is showing the model predictions. So the SIR model can do a pretty good job at explaining these complex biennial in some cases and non-biennial, for example, in Ohio, and also predicting outbreaks in Missouri and Utah, Colorado. And using this model, we were also able to predict that. So first, we found a large uncertainty in parameter estimates, but despite the uncertainty, we were able to predict that an outbreak in 2020 was likely. But that's not what happened. And COVID-19 emerged and everywhere went on lockdown, and so that prevented the transmission of many other respiratory pathogens, and that included enterovirus D68 as well. And so we didn't get an outbreak in 2020, but instead we got an outbreak in 2022. So one thing to mention is the BioFire stopped enterovirus D68 in surveillance in 2019. So this is now data coming out from New Vaccine Surveillance Network from, I want to say CDC, maybe not, I can't remember. But anyway, New Vaccine Surveillance Network, I think it's associated with CDC. Anyway, so then coming out from MVSN, and again, we were able to use this case data to capture the dynamics of the same SIR model to explain the post-pandemic dynamics of enterovirus C68 and try to predict what might happen after 2020, especially when now we're fully back to normal. But the problem is that we can actually get a very similar fit to this same data and get a vastly different predictions about what happens. And that points to the limitation of using case data alone. The problem is that case data only provides us information about who's infected, how many new infections are happening. So the SIR model has three compartments: people who are susceptible, infected, and recovered. But we can only observe infection, and this people who are susceptible and people who are immune are unobserved unless we have. So the problem is that this limited observation is going to lead us to large uncertainty in parameter estimates as well as long-term predictions and required vaccine coverage. But I think I don't have to talk how important serology is, but if we had serology, we could measure what proportion of people are susceptible and recover. In particular, we're interested in looking at age seroprevalence curve. This is data coming out from UK from a collaborator in Imperial College, Margarita Ponsalort. So if we could measure seroprevalence across different age groups and ask what proportion of individuals are susceptible, which is going to be the area above the curve, and what proportion of individuals of the population are going to be immune to future infection, roughly below the curve, area below the curve. And so to illustrate how we can actually integrate serosurveillance data on top of case surveillance data, let's start with the biennial epidemic. So it turns out that we can get a massive range of parameter estimates that are consistent with biennial epidemics. So R0 from 12 to 43. So this is saying one enterovirus infection could lead to either 12 new infections or 43 new infections, which is, again, wide uncertainty. But they all agree with biennial epidemic. And then once we have case data, we can try to narrow down some uncertainty. So that leads us to 20 to 40, and all of them can capture the observed pattern, post-pandemic pattern pretty well. But again, that's still a lot of uncertainty. The thing we can do is now for each of these curves, we can try to predict what age seroprevalence curve would look like. So the right-hand side, and then ask if we can measure enterovirus C68 serology, seroprevalence, that we can ask which of these curves are consistent with data. And this is where premise data comes in, where we have enterovirus, which provides us with enterovirus E68 serosurveillance from the US, and that allows us to pick out these curves that are consistent with both case data and serosurveillance data, which narrows down uncertainty a lot more now from 12 to 43 down to 18 to 27 in this case, and it's going to also narrow down our long-term predictions of what might happen. And there are lots of assumptions that also go into making this 2024 prediction. I'm not saying that's what's going to happen this year, but it's just an illustration of narrowing down the uncertainties. And we can also try to compare what proportion of the population is susceptible for each model trajectories, and compare that with, so previously, can I go back? I think I can go back. Oh, there. Yeah, so this is showing age zero prevalence curve, and then now we can turn this into by doing some extrapolating and averaging across population structure, we can compute a proportion of population that are susceptible to D68 infections, which is showing the 2 points in 2 points. So 2022 is the cohort 1, and the 2023 is cohort 1, but follow up longitudinally, and that also gives a pretty good agreement with the model predictions red. And so conclusion, mathematical models are quite useful for understanding these dynamics and making predictions, and combining case data and serosurveillance data can really help us understand better and narrow down parameter estimates and predictions. And people that I want to thank, Princeton University, NIH, Colorado, and lots of other collaborators and organizers. And thank you for inviting me.
Okay. Hi, everyone. It's a real pleasure to be here. I was trying to figure out what to talk about for this talk, because I take Jane as like the in-host guru from my perspective on modeling, and then Daniel's so amazing at statistics and population level modeling. So what I thought I would do is kind of just give you a taste of some of the work we've done in the context of COVID-19, and then just give a wish list of where it would have been nice to have more sero data, and sort of help contextualize sort of some of the use cases that I see. So I come at this with I still haven't figured out what button. Okay, I come at it with an engineering background, which sort of, and to convince you of all where that fits in, I sort of focus on the role that fluids play in transmission, and do sort of basic biophysical experiments understanding stuff related to transmission. So how do pathogens get in the first place, interact with the complex fluids that they meet, like mucus and other fluids, and then reach their targets. What happens outside the host when you emit these droplets? How do pathogens remain viable? And then I try to tie that into the models that we develop. So I'm glad Daniel already introduced SIR models. I don't have to do that. So firstly, in the context of sort of the in-host stuff that we do, I think a lot about mucus. Here I'm showing you sort of the role that it plays. It acts as a size and biochemical filter in the body that can exclude the passage of pathogens, whether they're bacterial or virus or what have you. But an important component of that picture is not just the proteins in mucus, but also secreted antibodies like IgA, which are around and can play a big role in impacting the mobility of pathogens. So for me, when I think about developing really explicit models for how does a pathogen, here's an SIR model at a cell level, right? And I'm sure Jane will talk about this more, but can we think about individual cells as being susceptible and getting infected, and model some of those initial dynamics? It matters to me how the virus gets around. It's sort of like how mobile an infectious person is in a population. So antibodies would change that picture. So this is not my work. This is actually from Brian Grenfell's team as well. This is Sinead Morris's work actually on measles. And here, this is just a nice example of that, showing an in-host picture of how having antibodies in your model would totally change the kinetics of how you'd expect a virus to proliferate. So my goal is to then connect those pictures to what we see at the population level. For instance, can I connect viral titers in a person to, let's say, transmissibility at the level of infection between people? So for me, it would be incredibly valuable to know things like, titer of antibodies over time in response to an infection, post-vaccination. IgA, maybe we think of as more of a mucosal antibody, but we can get signatures in peripheral blood as well. And maybe this is a bit of a wish list and beyond the scope of what we talk about with residual blood, but I think having the sort of longitudinal profiles and understanding these kinetics and being able to parameterize this more would be immensely valuable. So at the population level, I'll talk a little bit about some of the work we have done and are doing in the context of COVID. So starting with this picture that Daniel already explained, so that's great for me. One of the first questions we asked, and I think this work started in March or April 2020, was just, how's the waning immunity going to impact the dynamics of the pandemic? We used very, very simple models. They were not specific to a given region or really even reflective of any specific population structure. We just said, what if it's not SIR, which Daniel explained, but what if there's a possibility to revert to some level of susceptibility with waning? So we came up with, predictions, and so this is work with Brian Grenfell, which is how I know Daniel. We're both Grenfell alum, and then Chadi Sadroy, who's at Berkeley and who did a lot of this work with me. So we came up with maybe unsurprising predictions that if we don't have very good immunity that arises from infection, then we're probably going to see a lot more infections over time. Maybe that's not a big shocker, but we were trying to also come up with estimates for, clinical estimates. How could we, project things like burden of severe cases? And here, we are coming up with these estimates or profiles. So these purple colors, it's sort of a landscape of immunity in the population, if you will. So dark purple would be people that we assume have full protection. Light purple would be partial protection. But, and in theory, we could match this with serology data, right? But then we'd also need things like correlates of protection. So a huge thing would be to know, what is a tighter mean in terms of your likelihood of getting severe disease versus hospitalization versus, no infection at all. These would be hugely valuable things to have that are largely not able to come by at this point. Waning rates, with longitudinal studies, we're getting better ideas of that. This is at the time when we had no idea of that, so we were literally just coming up with numbers. There's also no vaccine at this time, so we were trying to come up with estimates for the role that would play. And obviously variation within the population would be great to get at as well. So the next thing we did was actually in the context of the emergence of vaccines, when all of a sudden we had vaccines that showed up way faster than we expected them to. I think we had made a best guess projection for about a year and a half from when we were doing that work, and then they popped up by the end of that year. So we were pleasantly surprised, and they were hugely successful tools. But then there was this multiple dose aspect, and we were told you should wait 8 weeks, but in Canada, we were sort of throwing that out the window, and we were saying, well, we'd rather just give more people 1 dose than actually give everyone 2 doses, whereas in the US, you were very strictly following the manufacturer guidelines, and we sort of wanted to do a bit of modeling on that and see what could the potential implications be of doing that. And from what I understand, those guidelines are really just based on the fact that when they ran the clinical trial, that's the spacing that they used. There wasn't, it's not like they'd run a study to figure out the ideal spacing time. They just said, we know it's okay if you do it like this, so let's just keep doing it like that. So here, for instance, if we'd had, data on responses after 1 dose or 2 doses, with what the different gaps would be, you can imagine, it would have allowed for much better projections of sort of the rollout for in cases like this. And then lastly, we did some work looking at sort of vaccine sharing. So we had we've developed these vaccines, and then we decided to just not give them to most of the world, which was an interesting move. So we just did some modeling looking at how, coupling regions and allowing for, in a very simple way, emergence of variants and things like that could allow us to make arguments not based on morals, although I think those arguments obviously exist, but epidemiological arguments for the benefits of doing this. And here, coming back to what we've heard a lot about this morning, I love the talks from the collaborators in Europe this morning, and hearing about the Global Observatory. Getting a better picture of global landscapes I think would be so helpful when you talk about, let's say, guiding vaccine strategy based on immune gaps and things like that, understanding which regions have been particularly hard hit with infection. It would be a hugely valuable tool. Right now, we've taken these super simple models that were not region specific, and we've tried to do some work with specifically in the Quebec context. So we've taken a compartmental model that had like 5 compartments, and I don't even know how many compartments we're at now. It's probably thousands of compartments, but we've tried to make it, really specific to capturing population structure, multiple strains, waning, cross vaccines, all sorts of things. So we have this compartmental model that I've developed with Mathieu, who's in the public health school here, and also my grad student and our collaborators at the INSPQ, Etienne. And we've been trying to use it to study counterfactual vaccine rollout. So if we've calibrated it to data in Quebec, you can see it does a good job. These are sort of the hospitalization data and then the model projections. the younger age groups, so there's just no hospitalizations to fit to, so that's why the curves look a little bit wonky there. And then what we've done is sort of use this model to say, well, what if we hadn't followed the scheme that historically was followed? What if we'd prioritized in different ways? Can we learn whether some would have been, whether we could have done better in terms of mitigating infection or hospitalization? So we get some interesting numbers. It looks like our results suggest that overall the approach taken was very good. We can sort of come close or maybe get better by a couple percentage points, but overall the strategy was effective. But we run into some real challenges. So these models have a lot of parameters in them that require, estimates related to transmissibility, that require estimates related to efficacy of vaccine against infection. And like Daniel was saying, when you don't have any idea of what the case numbers are in a population, there's really no way you can meaningfully come up with a value like that, because you just can't compare it with anything. So I can, we're more confident in our estimates, say, for protection against hospitalization, because then I can turn around and fit my hospitalization numbers against real data. We don't have, I could tell you it was really bad against transmission and there were 12 billion cases, but at the end of the day, I can't fit my cases against anything, so I can't really validate that. So that would be great. And we had a couple zero estimates taken from Quebec, but they were super, we had one data point and not a very representative time. So this is where that could be really, really effective. Okay, I just want to talk quickly about what we're doing in the context of HemaNet. So we've built this agent-based model that allows us to simulate disease transmission in a more structured way with this agent-based framework, which we've based off this CovaSim model, but it can account for multiple circulating pathogens. So this is my team of students who've been fantastic doing this, and really Nathan sort of brought all of this to my attention. He was an amazing grad student here who's now at BCG. But so what we're trying to do with HemaNet, and you heard Jonathan talk about this yesterday, is use this model to run basically in silico serosurveillance programs, and then look at the benefits we can get in terms of what we can learn about infection from what these simulations would tell us. So we're trying to implement that into these cost-benefit analyses to sort of run the programs without actually having to do it with those associated costs and come up with some of those numbers. So I'm over time. I'm going to stop, but happy to talk more about it later.
Thank you very much, Carolyn. And now we're going to move to Jane Heffernan, who is online, and I think you have her slides queued up. Oh, we have Jane and slides. Great. Jane, you're good to go. Can you hear us?
Sorry, I was just trying to find the unmute button. Can you hear me?
Yeah, we hear you just fine. Go ahead.
Okay, sorry. Thanks everyone for having me online. Sorry I couldn't travel. This is a very big teaching week for midterms, so I couldn't make it. But I'm going to talk a little bit about what we've been doing for COVID and what our main goal really is to tackle this question of immunoepidemiology. So we're really interested in trying to understand the distributions of immunity in populations and how this is driven by the history of infection and vaccination of individuals on their own. And so you can really think about, we're talking about vaccination programs, we're talking about the spread of infectious diseases, we're talking about serosurveillance. A lot of these things are happening at the population level where we have different studies. We have public health rollout programs of vaccines. And when we're talking about what we need to do or what the risk of an infection is in a population, we really do look at the population as a whole. But the population is made up of a lot of individuals. And so what we really need to do is try to understand, what the actual distribution of immunity is in the population and how that could change for the population that we're studying. So here I've got a picture of Toronto, just because that's where I'm based. But when we consider all of these things, we're also looking at how the makeup of the population is different. So you can imagine with different immigration rates and emigration rates out of Toronto, that this would be a very different population makeup compared to the population of Regina. And so we have to take all of that information into consideration. But when we are talking about distributions of immunity at the population level and risks of infection, we also have to take into consideration the characteristics that are different within each individual. And so these incorporate their age, their genetic makeup, their gender or sex. Lots of different characteristics can affect what an infectious disease is going to do when they're infected. But then when we take into consideration their immune response and their immune system, we then are able to get a good idea of what their susceptibility is. And this is from transmission from other people in the population to the individual. But then if they do get infected, so first we look at their protection against infection, and then if they do get infected, we have to look at their protection against severe infection, so symptoms, as well as try to quantify what their transmissibility is back to the population level. And so when we think about these things, we also have to think about how dynamic immunity is. We have to think about their effective immunity. And so we may, like in a surveillance or serosurvey, we may measure some anti-S antibody, but we know the antibodies are there, but then we have to use the modeling to quantify what protective capacity that anti-S antibodies are giving you in the body against what the current viruses are that are in circulation or different strains. And so when we're thinking about effective immunity, then we also have to consider, so we have infections that are jumping people up this conveyor belt, or vaccines that are jumping people up the conveyor belt, and they can happen at different times. But then effectively, immunity could wane over time. So we've got decay of antibodies and decay of T cell and B cells in the body. But the pathogen can also be evolving away from the immunity that you have. And so effectively, you're also just experiencing a decay in your effective immunity, even though you have antibodies and memory T cells in host. At the same time, at the population level, we're trying to understand what the protective levels of capacity against infection and severe infection are, given the different age groups. So here we take in-host models, we embed what we understand about what's happening in the body from infection and vaccination, and then we put this into large level SIR models. So thank you, the previous speakers were talking about SIRs, to look at what the levels of susceptibility are. So here we've got green is totally resistant, yellow is not totally resistant to infection, but has a very high probability of a mild infection if infected. Blue is less resistance to infection than yellow, and still a higher probability of a mild infection, but less than if you were in the yellow category. And red is totally susceptible, and the different shades here by age group. And then the white up here, the waves of COVID-19 infection over time. And the red curve, which is a curve, it looks like a straight line, but it is a curve, that's the hospitalizations. So we take what's happening in the body and we translate that to distributions of immunity in the population. So when we're thinking about what's happening in the body, we take studies of clinical trials for vaccines. We take infection studies from COVID-19 or other pathogens that we're considering in the lab. And we try to quantify what their immunity output is from infection and vaccination. So we have studies on AstraZeneca, Pfizer, and Moderna vaccines. We have mild, moderate, and severe infections, data that we fit our mathematical models to. And we track vaccinated cells, we track antibodies, we track the development of memory T cells, and et cetera over time. And then we project what their decay is going to look like, given some longitudinal data that we get from these studies as well. In infection type of model, we also have to consider how severe their symptoms are. And we use area under the curve to help us determine whether things are mild, moderate, or severe. But in all these cases, we get some measures of the immune response. Usually we have some interferon gamma, an interleukin, maybe we have granzyme B, and we get some measurement of antibodies that then we have to consider. So we take all of this information that we understand at the in-host level, then we have to embed on top of this mathematical models of behavior and NPI, social distancing behavior, and how that can uptake and wane over time as well, and the circulating strains. And then we use that to help us identify what the transmissibility is up to the population level, where we have this large SIR model that also keeps track of one doses, two doses, booster doses of vaccines by different age groups and different ethnicities and so on. So when we're doing all of this work in-host, we also do take in consideration gender, comorbidity status, where the people are living, even their occupation, to try to determine or to identify what the outputs in their immunity will look like and how that is defined by the way that they live, they work, their family size, and so on as well. So this is just giving you some things that we can look at. So when we're looking at in-host modeling, we do some similar things to what Carolyn was talking about, where we do in other projects, we've looked at separating doses by more than 21 and 28 days to see what the output of immunity is. Here, what I'm showing is just we've fit a model to Pfizer and Moderna data where we had 21 and 28 day differences between the two doses. And then we looked at what their decay in antibodies would be to suggest when a third dose or a booster dose would be needed. But over all of these studies that have in-host data, what we try to do is we try to project longevity of immunity from vaccination and infection. And a lot of these studies don't have measurements of T cells. So what we try to do is when we have information on some other components of the immune response and antibodies, we try to project what the T cell immunity is going to look like. So that's going to be important when we're looking at protection from severe infections in the future. So when we're looking at immunity distributions, we look at different scenarios as well. You can see here, so we have like the beginning of COVID-19, and this is calibrated to hospitalization data for Ontario, but we do this for many different provinces. But you see that the beginning of the pandemic here, and then we have our uptake of vaccination and our booster doses. So we have people shifting from lower levels of immunity to higher levels. But then when we had Omicron sweep through the population, we had to translate the effective level of immunity in the population down because people who are vaccinated could also get infected with Omicron. And then, so this is just an example where, you know, after a certain date where we have good statistical calibration, we can then use our distributions of immunity model to project forward using different scenarios of what, you know, a future wave of infection would be. And so this is work that we did in early 2023 to see what spring and summer and fall might be looking at over time. When we do these calculations of what the waves of infection will look like, the models can also give us an idea of what mild, moderate, and severe infections look like by age. And so here we have blue are mild infections, and the lighter colors again are the younger age groups. The darker colors are the older. Yellow are moderate infections, which show symptoms. And in the red, we have individuals that have to go to present to doctor. And then some fraction of the red then go to hospital. So what's really interesting about this is that we can use this type of modeling structure to project forward what our outputs of infection will look like in consecutive waves of COVID-19 or any pathogen that we're looking at, which can then help inform what rollouts should look like for booster doses of vaccines and to also provide some information to impact public health, social distancing policies or recommendations like mask wearing as well. Talking about seroprevalence data, we've been mostly talking about effective immunity in this presentation, but seroprevalence data and serosurveillance enables us to reverse engineer what immunity will look like. And so this is a similar graph, but instead of effective immunity, we're looking at seroprevalence, and we've been able to compare our output of our age distribution model for COVID-19 to actual seroprevalence data from the CBS, courtesy of the CITF. At the same time, here's the ABC study output pre and post Omicron. And when we track our infections in individuals that are not vaccinated versus vaccinated, and whether they're coming from booster dose compartments or after they've only had one or two doses, we're able to compare the output of our simulations to the ABC data, and we get distributions that are quite similar to what are measured here. Just before I finish, I wanted to highlight CBRF proposal that Dr. Carolyn Collane and I co-PI'd for the current CBRF competition. This is called sim.net. What we have are statistical, dynamic, and economics modelers from across the country who use modeling to tackle different components of the biomanufacturing process. And so related to HemaNet, we're looking at surveillance and the integration of seroprevalence and serosurveillance data into our population level models, but also our individual level infection models. But when we're thinking about generation of immunity, we can also look at this information down here at the lab, where at the time that we're looking at animal models and at the bench, we can use in-host modeling and in vitro and in vivo modeling, if you will, to look at how immunity is generated and then how that can translate to humans in clinical trials and surveillance expectations. So I just want to thank the large number of collaborators that I've had on these projects, and thank you to everyone for having me here.
Great, thank you very much, Jane, and thank you to all of our panelists. We've now got a few minutes here for questions. So those who have questions, please go up to the mic and or online, and I'll bring your questions forward as they come. And I have a few questions to start out, though, so just to get us going. Oh, we have a question right there. Please, go ahead. No, it's okay. I've got lots of questions, so we'd rather start with yours, actually.
Okay, I just have one question. So you talked about, like really a very exciting presentation about modeling for COVID and, for D68. And what I'm wondering about, we talk about multiplexing panels for serosurveillance. We talked about like triple-demic, outbreaks last year over the fall. Are there equivalents like multiplexed models that take into account, multiple, circulating pathogens and different levels of immunity to them?
Start and come back. Go ahead, Carol.
Yeah, so this agent,
It's working.
the ABM framework that I bring it closer to you, maybe. The modeling framework that I ran out of time to talk about at the end, I said we had adapted it from another agent-based model, and the way that we adapted it was really to make it multi-strain. So it's N-strain co-circulation. And for us, that's really interesting because from what we can tell, there's lots of studies that look at, let's say, timing vaccine rollout with whether an epidemic is just starting, whether, sort of where on an infection curve you are. And so having the ability to think, like, for instance, it intrigues me to think about, well, what if a new pathogen comes in when you're in the middle of an influenza outbreak, or if you're, depending on other circulation, and then what happens if there's cross immunity in any way. So the challenge we run into is it's really not obvious to parameterize these models with, like, is there really good data on what's the delta and transmissibility if you have, flu and RSV at the same time or something, so some of these like exact parameter estimates get a little tricky, but this is something that we, that in theory we have the capacity to do, and we're trying to do some initial simulations on that. But I think you're right, and especially like when you talk about the triple-demic, and right now we're so focused on these 3 respiratory pathogens, I think thinking about them in concert is going to be important.
Thanks, Carol. Sang-Woo, do you have a comment to add? Or?
I guess it also depends on, first it depends on the question, but also I guess depends on what kind of pathogens you're targeting. Like if you have a multiplex thing that can test for RSV and D68, that's great, but I guess the interactions are going to be so weak that I'm not sure if it's worth the computational effort to try to model them at the same time when, obviously if we're trying to understand something like flu A, flu B, or H1, H3, something that we know has a lot of interaction, then we want to be able to test them simultaneously and then model them simultaneously. But if that's not the case, and so I think it really depends on. But there are, I guess, it depends on the question, depends on pathogens. But there are, like Caroline said, there are lots of models that do offer multi-pathogen inferences, and yeah.
Thank you, Jane. Anything you want to add?
Yeah, I think one thing that we look at is the protection effects of the innate immune response that are heightened while you're infected with something else. and how that can provide some protection against infection from other things, and how this can change with age as well. The interesting things about but this is that cross immunity and just even having the innate immune response can give you something where you have an oscillatory output of other infections. So if we're considering COVID and RSV and flu, one can, like, you have longer diseases, shorter diseases, different immune responses, including the innate immune response. And so you can induce periodic, like a oscillation in your outbreak of one just because you have coexistence of the other in the population. So those are really neat. But we do try to understand what happens in host when you have pathogens that are interacting, or you've been exposed to one pathogen after the other. And I do have some work on influenza with different strains, where we infect a ferret with one strain and then we challenge them with another one so many days later to try to quantify what the protective effects of the innate immune response, the B cells, the humoral immunity, and the cell-mediated immunity are over time.
Great, thank you. Okay, interesting. Next question, please.
Yeah, thank you. Thanks for that all of the incredible talks. So my question is to all the panelists. In the spirit of us sort of discussing establishment of a network during the so-called peacetime that doesn't exist, as we've mentioned, can you comment on how you were able to, during the pandemic, rapidly share back this type of data to public health? And how maybe those structures were or weren't sustainable, and what needs to be put in place so that going forward, that sort of sharing can occur?
Any takers? Sangwoo, you look like you're about to speak. Okay, Jane, we'll put you next. Go ahead, Sangwoo.
I think we had trouble with a lot of data sharing too. A lot of them has to go through a lot of steps. You have to get approval from a lot of people. So I ended up just scraping a lot of publicly available data. So there's a lot of cases where data that's available, publication, but they're not willing to give you numbers for whatever reasons. So I would just have to dig in manually, use some kind of tools to try to get the numbers out of figures. And so I'm not sure. I don't know if there is a really good. I don't know. I don't know. That's just my experience. I had a lot of frustration with that, but I don't really have a good solution or suggestion for it.
Yeah, I've heard that called data raccooning. Yeah. Do you have, how on the output side, did you have any experience with your results from your work and translating that into decision makers' hands?
I think, I mean, yes, I worked, so I spent a lot of time working on guiding isolation testing guidelines for Princeton University. And so with those people, we met frequently, and then I knew exactly what numbers they wanted. So, I mean, I think it comes down to communication issue, where I'm not going to show them what each model projection compartment is. They want to know, how many isolation beds do you need? How many cases are they expecting? And so I think it comes down to how well you communicate, but that also doesn't translate very well with I think there was good communication between me and the decision makers in the university, but the university students had no idea I was doing this modeling or what kind of things were going under the hood. So there is, again, a transparency issue for a broader public, and yeah, that's an issue too.
Good point, especially when the models become more complex. And yeah. Jane, did you have anything to add to this?
Yep. So over COVID-19 and not post COVID-19, we're still in it, but post the severe components of COVID-19, my modeling group was involved in lots of different aspects for informing governments and decision makers, as well as trying to get data and different versions of data. So it could be even that, we could get a spreadsheet of data, but if we only had a certain level of approval, we'd get the PDF of the spreadsheet. So then we would have to scrape the PDF to get the actual data points. So there was lots of data scraping coming from my group as well. But at the beginning of the pandemic and throughout, my group was involved a lot in healthcare demand modeling. And so we modeled the demand and the ICU length of stay and ward bed length of stay in all of the provinces. And we wrote a weekly report to Health Canada to provide to the upper offices of the federal government At the same time, we also were involved in some informing of the science table in Ontario and the new science table in Ontario too. I can't remember what the acronym is for that. But I think that there was data issues aside, and we can have just a whole panel just on data issues. There was a lot of openness to being able to share data. Data sharing agreements then took forever to get. And so a lot of the questions that were posed where you needed the answer yesterday took months to get the data. So then you would have to be answering questions 6 months behind. So there are a lot of assumptions, and Carolyn alluded to this, a lot of assumptions that go into models that we then hope that, later on we can test some of those assumptions. But, we're pretty lucky that we have lots of great modelers in Canada and around the world that embedded lots of assumptions into our models, and some of them were very robust, and we all learned from each other on online meetings and conferences to inform our models as well. So I think a lot of the modeling that happened here was very good and robust.
Thanks, Jane. Anything, Carolyn?
Yeah, I mean, in some ways we were lucky with some of the work we were doing because we were hiding behind this veil of keeping it so general and not being specific. But when we spoke to WHO and SAGE, and we, especially about the vaccine sharing stuff, and I was struck by sort of just like, I, as a modeler, I sort of hide behind all the caveats that go into the model and all the limitations, and they would be like, well, give us a number, like, how many, what do you have to share, or what's the duration of immunity? And I guess coming back to what Jane's saying, there is uncertainty in these things that make it, difficult, especially without the right data to validate against. So that was, I found a tension was, how can you be confident enough in the numbers you're giving, given all the uncertainties are embedded, but still given a number that's actually useful in some way. In terms of the calibration work we're doing, running into similar hurdles, getting that hospitalization data from Quebec was not a trivial process. So it seems like maybe facilitating that in some way moving forward will be beneficial. But as Jane alluded to, for me, I came back. I started my job like basically during the pandemic, and a lot of things were challenging, but one really amazing network that came out of it was this CanMod network of Canadian disease modelers, and Jane's part of it, and we had a meeting a couple months ago. But I think like Jane's saying, there's so much willingness to share a methodology, to share data, to share insight, and that's so positive. I mean, I'm speaking in a Canadian context, but I think that goes more broadly than Canada. So, and I don't know what the answers are. I'm not in this sphere, but just finding a way to make these collaborations and data availability easier, I think would have such a big impact in the future.
Great. Alton, did you have a question?
Yes, thank you. Yeah, so I as someone who does some decision analytic modeling, I definitely relate to the kind of being a data scavenger and taking what you can get. I think it's not really that well appreciated how mathematical models can help quantify the uncertainty and quantify which data elements would reduce the uncertainty in the models. So I'm curious the extent to which have you been able to date to try and use the models to show like this is the data we should collect, so that we're not just kind of coming at the end and picking up the pieces we can get, but we are part of the conversations from the beginning about what data should be collected. We can help direct that in order to basically spend the money we have to get the data that we need to make better decisions to improve health. I think modelers have a role to play there. I'm curious what you think about that and how we could make that part of the process where we're kind of coming in earlier.
Great question, Alton. Before you go, because that was one of the areas I wanted to dive into, so I'm just going to add a little slight addition to that. So maybe if each of the panelists could tell us, as Alton said, what's your thought on that, being able to, what's the value of information essentially for you in your models, particularly the ones that you're missing, and maybe say a little bit about like what would that thing have to look like? Is it a single parameter, or does that have to be data linked to something else? So while you're thinking, does anyone want to go first?
I can.
Okay, go ahead.
First, I think it's a really great question because I think models allow us to simulate synthetic data, and potential scenarios, and that allows us to generate what data might look like and test what kind of data would give us what kind of information, help us make better predictions. And so I have actually done, for setting up premise, I've done power analyses, not some like standard t-test or some sample size calculation, but you can use models that are grounded in past previous parameter estimates and predict what kind of data would look like, what your prevalence curves would look like depending on different scenarios, and ask if we had scenario A, B, C, how much data would we need for if we had different, say, say having what's the value of having two separate cross-sectional data versus having one cross-sectional data and the longitudinal data. And I think that's where really modeling becomes really powerful because so yes, you can do that. I guess that's.
So you can do it, but there's no general answer to the type of data that you're really thinking.
No, I think it really depends on really the question you're answering. For example, if you want to just estimate, say, population level dynamics, longitudinal data doesn't actually help as much as cross-sectional, getting another cross-sectional data. Cross-sectional data is just as good as getting longitudinal data. I have simulations to back that up, presented that last week actually, but obviously if you want to look at individual level antibody or titer kinetics or things like that, cross-sectional data is just not going to give you any information on the dynamics at the individual level, and you absolutely need cross-sectional and longitudinal data. And so I think it really depends on the kinds of question you're trying to ask, and there's not going to be a general answer, but I think the general answer is more data the better.
Carolyn? Jane, we'll come back to you. Sorry, go ahead, Carolyn.
Yeah, I think, Alton, that's a great question. I guess like where we're immediately running into it right now is in this Quebec study where we just get estimates for things related to infection and transmission, and we don't really know what to do with them because we don't know case numbers. So I was actually talking to Mike earlier, like having young children who are always sick. I'm just like, to me, it's so obvious why we should just have like multiplex assays for everything, because I feel like cheated by not knowing like which virus is it, what protection do we have currently, who else is going to get sick next. So I don't know, like could I imagine a world where we could, actually have like in-host kinetics following, let's say, infection versus vaccination versus what if you've had an infection and then you're vaccinated. But, and when these data come in, like over the course of the pandemic, obviously these data sources are generated, but, maybe as you're trying to come up with some of these projections and do these models initially, these are sort of questions and data sources that would be so valuable. Sometimes we did some work on the potential utility of wearable sensors, which would pick up physiological signals and maybe give you messaging like you might have an infection, potentially quarantine or something like that. And there I was just and there's clinicians here in the room who could probably say more about this than I could, but I was so struck by this idea of, do we know even enough to say, if you're infected with flu versus RSV versus COVID, could we expect that your, immune response would be different enough that your physiological response would have different signatures for the different pathogens? Could you even expect that, if you're measuring heart rate or whatever other metric, that you could tease apart different infections? So I think there's cool things that we could generate even in a non-pandemic context. We can't say peacetime anymore after the talk this morning, but I guess that's sort of some of the things that come to mind on a fantasy list of data.
Thanks, Carol. Jane, over to you.
Yeah, so thinking about in-host modeling, we love to be incorporated into the picture before the actual study occurs, so in the study design, so that we can help the researchers determine if you can only get 4 measurements, what dates or what times do we need? We certainly need a very early one, we need a late one, and in between, what kind of optimization can we do to help you, design your study so you can get ultimate optimization of output, so that we can also help you with our modeling studies inform compartments that you're not measuring. And so we can do that at the bench, in animal studies like rats, hamsters, ferrets. Think about the clinical trials. When should you take your measurements? How long should your study be? How big should your cohort be if you want to answer these questions X, Y, and Z? So that's something that we've discussed with sim.net, which was that CBRF proposal that I posted at the end. But in terms of thinking about other components, we could think about, what if you can't measure something? So we can help determine what the uncertainty is in that data missing. So if you have missing data and you can never measure it, then how good can our actual outputs ever be? Maybe you can only get to this level of uncertainty and you can't get better just because you have that missing data. And then if you think about in terms of longitudinal versus cross-sectional data, there's benefits to both. if you can get like both, that would be great at the same time, cross-sectional with longitudinal. But the more data, the better, but we are restricted with funding levels of our research. And so we really need to like a big, health economic study of all modeling and all CIHR studies to see, really what can we do with $500,000 or for a math grant, what can we do with $15,000 a year
Okay.
from NSERC?
Good policy on, comment on research policy. Thanks, Jane. Okay, Tim, last question to you.
Very quickly, I'm just wondering, sometimes when you're doing these models, you sort of run into roadblocks because some of the parameters you want to estimate we don't really know the answer. So you're sort of in the dark, and it's not a function that you can't make an estimate, it's just that we don't know enough about basic immunity, right? And so I'm just wondering, do you have a feedback mechanism in your models, which is going back to the immune experts and saying, do you know anything about these things? Or if you don't, do they represent reasonable research priorities that we should be directing more fundamental research towards?
Jane, do you want to start maybe this time? Do you have anything to say?
Yes, I have lots to say, but I'll keep it short.
Yeah, we only have a minute and a half, so a quick thought. Yeah.
So yeah, we work with immunologists. And so often, and this is related to the previous question, often they give us data, we do some modeling, and we say, hey, if you had this other data, we could tell you this, and this as well. And they're like, oh, I have that data. We can give you that data too. But sometimes they don't have the data. But we do, we have very close collaborations with the immunologists and epidemiologists and people in public health and biology. And we really try to have this integrated network of, our projects going on, and then feedback to the, to the people in the other field, and see if we can help design a study to measure these things that we don't know.
Thanks. Any other thoughts from the crowd?
Yeah, I mean, we encountered that in the early COVID work, and I guess our approach was just as we got more information, we we started out in with massive estimates on waning rates. We refined those as we got more information. But yeah, I think working with that measles diagram I showed, it's actually from an amazing paper where it was an animal study, and they had the most incredible data of T cells, memory T cells over time, virus titers over time, antibody levels over time. If you could and this is an animal model, but if you could have data of that level and on large amounts of people, it would be incredible from a modeling perspective. So obviously working with people, in the clinical level, immunologists is hugely valuable in that respect.
I think having more data is great, and I think we can also use models to predict what might happen in different scenarios. What if waning was fast, or what if waning was strong? That's a lot of things that we had to do with, even for a lot of COVID responses, that we had to make assumptions. But I think that's the power of modeling, that we know as long as we know what assumption we're making, and as long as we're clear about it, then we can try to understand what outcomes are possible for different assumptions. Or if you're a little bit fancier, you can put Bayesian priors on it and then integrate across different uncertainty, but that's a side note.
Great. Well, good point to end on. There are some limits to our knowledge. And I'd like you all to join me in thanking the panelists.